Hello everyone. Today we are going to see what is pyrosequencing. So pyrosequencing is one of the methods which you can consider as your next generation sequencing methods. So in the last lectures when we were discussing about Sanger's method and Maxim-Gilbert method, I had told you that your electrophoresis, when you are performing electrophoresis, a major disadvantage is sometimes the bands may not properly resolve. And in that case you will be not able to count some of the nucleotides, miss upon some of the nucleotides, and the sequence that you will get would be wrong. So pyrosequencing is one of the method which was developed by a Swedish company, and it is combination of your light emitting luciferase reaction with your PCR based reaction, and the PCR here is your oil immersion PCR that is being used. Okay, so pyros is fire, so that stands for whatever the light that is being generated. Okay, and sequencing is finding out the sequence of the DNA. So luciferase enzyme is usually been found in the nature. You can see here that either you can find it in your fireflies. Okay, everyone knows it, we call it jugnu in Hindi. So fireflies, and this is a jack-o'-lantern mushroom which also lights up because of this reaction of luciferase. So let us now see about the pyrosequencing biochemistry, what it is like. So we all know that in DNA sequencing or synthesis, whenever, because it is a PCR-based method, DNA synthesis would be taking place. So in DNA synthesis, whenever a DNTP is added to the DNA, what happens is, a primer is used, and this primer would be having this free 3' OH group, to which the incoming DNTP would be added. So when the incoming DNTP is added, what will happen is a monophosphate group. So DNP will be getting added to the existing DNA chain, and an inorganic pyrophosphate would be released. So in this reaction, this particular pyrophosphate is what actually is going to be measured. So that is what a DNTP is attached to the 3' end of the growing DNA strand, and the two phosphates on the end are released as your pyrophosphate. So you have the next enzyme now taking or involved in this reaction. You have ATP sulfurylase. So this ATP sulfurylase will be using this PPI, that is your inorganic pyrophosphate, and another substrate, that is your adenosine 5' phosphosulfate, to make ATP. Okay, so that is the next reaction that happens. So you can see here APS along with PPI that will react in presence of sulfurylase to give you the next product, that is your ATP. Next is luciferase enzyme, and that causes these fireflies to glow. Okay, so the same enzyme, it can utilize your luciferin and ATP as your substrate, and it will convert your luciferin to oxyluciferin. And this oxyluciferin is the one which is going to produce this light, and that light can be captured as so that light would be generated as a peak. Here you can see that it will generate as a peak. So whenever a peak is added, you understand that a particular nucleotide has been added to the chain. So this amount of light, whatever is being released, is always proportional to the number of nucleotides that is being added to the new DNA. So that means whenever one DNTP is getting added, what will happen is only one PPI is going to be released, and that one PPI with one molecule of adenosulfate will be producing only one ATP, and that will be able to produce only that much amount of oxyluciferin. So only that much. So the amount of ATP is going to be say how much amount of light is going to be generated, and the amount of ATP that is being produced is dependent upon how many DNTPs are added. So if you have 2 or 3 DNTPs added together, what will happen is the number of pyrophosphates that are being released are going to be more. And finally you have apirase, which is going to destroy whatever the remaining DNTPs are there. So these are the main reactions that actually take place during your pyrosequencing. Okay, so you can see here that any DNTP that is remaining back, that would be broken down into your NDP, that is your diphosphate, and your MP plus your phosphate. That is how the breaking down would be taking place. So once it has been broken down into diphosphate or your monophosphate, it is not in a condition to be added to the growing DNA chain. So let us now see the general working scheme of this particular method. So first of all, what happens is whatever the DNA that we need to sequence, that DNA needs to be amplified with PCR. Now, usually in a PCR, we will have a forward primer and a reverse primer. Here also we will have one set of primers, forward and reverse, but one of those primers would be labeled by biotin. Now, we all know once you are having, so when one of the set of primers are labeled by your biotin, when you are getting the product after the PCR is being done, so there would be always one strand out of the duplex which would be labeled by or which would be bound to biotin. So now when you are adding your streptavidin bound beads, we know that biotin has an affinity towards streptavidin. So whenever you are adding these beads, this biotin labeled whatever your strand is there, that will get bound to this streptavidin bound beads. So now what will happen is when you are using NOH is known to denature the DNA. So this duplex will get denatured and the template and the strand which is being labeled with your biotin, that remains bound with the streptavidin. You can see over here. So this is your bead. This is the streptavidin which is being bound to this particular bead. And here you can see this is your PCR product, the template which is being bound to the biotin. So this product would be used as the template for the sequencing method. Okay, so in the next steps, whenever you are adding the primer, it would be going and binding to this particular chain which is bound to your biotin. So now whatever the template that has been used, so this template would be pipetted out in the microtiter plates, okay, and then all the requirements would be added. So you will be adding the sequencing primers, the substrate mixture. So substrate mixture will contain all the substrate that are required, which we have already seen in the pyrosequencing biochemistry. So your APS, luciferin, all the enzymes, that is your DNA polymerase, sulfurylase, luciferase, apirase, everything is going to be added. Now, one very important thing that we have to see in this case is, like every time here, so you won't be having 4 different reaction mixtures that are being working together. It is not that way. Now, you would be having only one single plate, okay, a bigger plate in which the reaction continues. Now, what happens is at a time, only one DDATP or one GGATP is going to be present. Okay, so here you can see that the next nucleotide that needs to be added should be complementary to T. So the next nucleotide that should be added should be your DDATP. So now in the tube, one by one, this nucleotides, your DDATPs, would be added. So when your DDATP is added, it will go and bind, and what will happen is there would be the PPI that is going to be released and the light would be emitted. But in the same case, if you are adding DDGTP, there would be no insertion and the PPI will not be added, so the reaction will not be completed and the light will not be coming out. So light will not be emitted. So the preparation is placed in the machine. The primer is first hybridized on the template, and this is followed by your gradual DNA sequencing. So now what happens is the machine adds one of the 4 necessary nucleotides. So that is what I am saying, one by one, the nucleotide triphosphates are going to be added. It is not that all the nucleotides are going to be added at the same time. So if the DNTP is added, because it is the complementary to the next base of the template, it will be inserted and the pyrophosphate is going to be released, and the entire reaction that we had seen, that would be done, and the oxyluciferin would be produced and the light would be produced. Okay, so and if it is not been inserted, then the PPI will not be produced and the light will not be emitted. So at the end you will see here that the light production is recorded during the entire procedure using a camera which is quantified, evaluated and present in the form of peaks. So you can see the peaks over here. You can see when you are having a single nucleotide that is added. So A, this is for A, this is for G. So now what happens is you can see here that this peak is very long and this peak is small. So what does that suggest? That at a time there were 4 Gs that were added when your DDGTP was there, and so that was the reason why 4 PPIs were released, and because of that the light that was generated was more. That is why we had seen in the first slide itself that the light is whatever the light is emitted, that is going to be proportional to how many nucleotides are being added. So whenever you are going to get, so you can see here that this peak is 3 times this peak. So that means in that particular case there would be 3 T's are there. Here you can see 1 C is there. This is corresponding to 2G. Okay. So because of adding of your nucleotides, wherever you are getting your peaks, you can see you will be you can see that that is going to be your sequence. So this is your sequence. You have A, then 4 G's, then you have T, then 2G, then C, then 3T, you have 4G, 2G. So this peak, depending upon the peak whatever you are getting, so that is the sequence that you are going to be loading, so or counting. Okay, so if no nucleotides is inserted, it will remain pitch black in the well. To remove the surplus DNTPs, we have already said, so one by one this DNTPs have added. So when you have added your DATP, the reaction has been completed. Now what is to be done is you have to remove all whatever the remaining DDATPs are there. So that would be done by your apirase. Okay, so that is the purpose of your apirase, why it is going to be utilized. So let us now see what is the advantage and disadvantages of this particular method. So advantages, it is a rapid method for analyzing of individual base polymorphism, because see what happens is here we are seeing very small structures of DNA. Okay, so very clearly we can see if there is any kind of SNP also, that can also be very nicely analyzed by this through a high throughput test. Okay, so up to 4,500 tests can be analyzed, and with a second related system you can sequence up to 48,000 probes per day. Okay, so the run time is very fast. Okay, it is accurate. Parallel processing can be done. That means at a same run you can have many different your DNA sequences, you can run it at the same time. Okay, so then easily automated, all these things are automated, and that is the reason why the run time is very less. And very important thing, you don't require any gel electrophoresis. Now let us see the disadvantages. It is not a procedure for simple research laboratories because it is an automated technique. Definitely you will be requiring a proper technology, a proper machine for this. So small labs like us, we cannot go for this. The acquisition of such a system is profitable only when you are the routine laboratories in such a large quantity of standardized analysis. That means like if in our lab we are in a daily basis we are doing your sequencing method, then only it makes sense that we buy this particular system. Okay, so for example, where you will use that, so there are some labs which do the characterization of microorganisms or diagnosis of very different diseases, which that is your genetic diseases, or your SNP studies and all is being done. Allelic frequencies are being analyzed on a daily basis. For such kind of labs this can be possible, and the work requires is considerable and special software is required. Okay, so instruments and everything, a special setup is required for that, and the cost is around $130 to $2 lakh, and an attempt to analyze an SNP for instance costs about $1.25. Okay, so it is more economic that we outsource these kind of your sequencing methods instead of buying a sequencer just for analyzing 1 or 2 sequences. Okay, so this is all about the basics of your pyrosequencing. We have seen what is the biochemistry behind the pyrosequencing method. We have seen what is the general flow. In the next video, I will be talking about specifically about the technology that was being developed. Okay, so in the machine, how the actual reaction takes place, that would be discussed in the next video. Thank you.